All right, guys, let's get busy talking about our disorders of sexual development, also called DSDs. We're going to have our master diagram again here, and I want to talk about how we're going to approach today's lesson. So in all of our round bullet points here, we're going to be discussing important pathophysiology, and then we're going to go through each of these square bullets, and each of these square bullets are going to be a physical exam finding or a structure, and we're going to discuss the embryological time points and the actions that lead to the presence or absence or function or non-function of these parts respectively. I want to start out by talking about 5-alpha reductase deficiency. Now, if you remember, 5-alpha reductase is going to be an important enzyme, and it's going to exist down here in our diagram, and its job is to convert testosterone to dihydrotestosterone, also called DHT. So if we have this enzyme not working properly, we are going to get failure of testosterone to convert to DHT. This means we're going to have increased levels of testosterone because it's not getting converted, and we're going to have decreased levels of dihydrotestosterone because there's none of it coming from testosterone. Now, if you remember, DHT has a really important role in developing the sexual characteristics of males with XY chromosomes. So what happens with DHT is responsible for the development of the external genitalia. And so when our DHT is absent, this is going to lead to ambiguous male genitalia. So let's go ahead and review here. We're having a decreased conversion of testosterone because 5-alpha reductase is not working. And so our DHT is going to be decreased, and then we're going to have ambiguous genitalia because the genitalia rely on DHT in order to show the male phenotype. So this is a great opportunity for us to review some of our genital embryology. We're going to have absence of phallus elongation, and you'll remember that the phallus elongation is going to occur at the genital tubercle. And I'm going to highlight this for you guys here in this red area. This is the area of the phallus that extends from the genital tubercle. Moving on now to our next structure, we're going to have a bifid scrotum. And you'll remember that this is due to improper development of the labioscrotal swelling. And I'm going to go ahead and highlight this in our diagram here, which is going to be this area down here. And because of this happening, we're going to get this bifid appearing scrotum, which is going to look like it kind of has two pieces. So it's going to kind of look like this, whereas normally it should look as one uniform pouch. Next, we're going to have hypospadias, and hypospadias, if you remember from a previous video, is going to be a hole where the urethra is popping out of the penis on the ventral side, and this is because we have failure of the urogenital fold here in this case. And I'm going to go ahead and highlight this area in gold here. And then it's also important to remember that DHT is going to be responsible for proper development of the prostate, but the prostate is not an external structure, so this will not be seen on physical exam, but I still wanted you guys to know this. And then it's important to note that these symptoms are going to last until puberty. So we're going to have these ambiguous genitalia until puberty, at which point testosterone levels will really ramp up, and the testosterone is going to be enough to start changing some of these external genitalia. Whereas in the beginning, we needed DHT because DHT is like mega potent. So just a small amount of DHT is enough during development to cause these male external genitalia to form. In terms of our lab workup, we're going to see that the testosterone to DHT ratio is greater than 20. And this makes sense because if we remember here, we have increased levels of testosterone and decreased levels of DHT. And this is because 5-alpha reductase isn't working. And that's how we get this ratio of greater than 20. And just to review, we are dealing with people with an XY karyotype, and they are also going to have normal estrogen levels here because there is nothing affecting estrogen. We actually have normal levels of testosterone at baseline, and so there's not an excess of testosterone that's being converted into estrogen. It's just that our ratio of testosterone to DHT is off in these patients. I want to quickly mention that this is going to be an autosomal recessive condition. Sometimes they like to ask about genetics on test day, and I want to have you guys covered there. Let's move on now to talk about androgen insensitivity syndrome, and I think this is a very complicated disease process, and it covers just about every portion of our diagram. So I think if you can understand this pathology, you have a really good grasp on our genital embryology diagram. So let's go ahead and get into it. We're going to have XY karyotype in these individuals. However, what is happening here in terms of pathology is that we're going to have a defect in our androgen receptor, making it insensitive to androgens. So when we think of androgens, we're going to think of things like testosterone. So here, if I was to draw a little androgen receptor like here, and then We have testosterone, which I'm going to represent with a T. Basically, the testosterone cannot act here on this receptor because it is genetically modified into a way that it can no longer interact with testosterone. It is important to note, however, that these patients are XY, so they have a Y chromosome. And we know that our Y chromosome has the SRY gene, which is going to produce testes determining factor. So these patients will go on to grow testes just fine. And we know that testes are going to be composed of Leydig cells and Sertoli cells. So to kind of orient ourselves in our diagram here, our undifferentiated gonad went to testes because we had a Y chromosome that was expressing testes determining factor. So we got testes, and therefore we're going to have Leydig cells and we're going to have Sertoli cells. And those cells inherently have nothing wrong with them. So our Leydig cells are indeed producing testosterone, and our Sertoli cells are going to make anti-Müllerian hormone just fine. So let's talk a little bit about what happens in these two situations. So since we have production of anti-Müllerian hormone, it's going to come over here and it's going to cause regression of our paramesonephric ducts. And because we have no paramesonephric ducts, we are not going to develop structures like an oviduct, a uterus, or an upper vagina. And that's going to be important a little bit later. Now, also, we will have no mesonephric ducts. And this is because the mesonephric ducts are under the control of testosterone. This testosterone is unable to have effect on the mesonephric ducts. It cannot support them because that receptor, which we talked about earlier here, is not working. Therefore, the mesonephric ducts do not get the hormonal support that they need, and they are going to regress. Therefore, we have no mesonephric ducts and no mesonephric duct structures in these individuals. Let's talk a little bit about how this presentation is going to occur. We're going to work our way through our square bullets and explain each one of them. So these patients are usually female presenting, and this is because they don't have the effect of any strong androgens like testosterone, so they're not going to get any heavy masculinization that we would expect with testosterone agonism. Next, let's talk about the fact that they have a rudimentary vagina. So if we look down here, when we talked about the fact that the paramesonephric ducts are going to regress because we had formation of anti-Müllerian hormone, that means that our upper vagina is not going to be forming. And so the rudimentary vagina is just the fact that they don't have a mature upper vagina. Additionally, we're going to see primary amenorrhea. And why is this? Well, in order to have a menstrual cycle, you need to have a uterus. And if we look at our diagram here with the AMH leading to absence of paramesonephric ducts, we're not going to have a uterus. So in order to have a menstrual cycle, you need to have a uterus. These patients are not capable of having a menstrual cycle. Next on our list is going to be cryptorchidism. And why is it that these patients get cryptorchidism? And the reason here is because our testes are reliant on testosterone in their migration from the retroperitoneum down into the scrotal sac. And so if this migration does not occur, you're going to have testes somewhere in the retroperitoneum. Sometimes they can be found in the labial folds. They're not going to be in the correct position. And it's important that these patients get treated with orchiopexy because remember, anytime we have testes that are not in the scrotum, they are at risk for developing into cancer. Next on our list is going to be our absence of mesonephric duct structures. And why is this happening? Well, we know that the mesonephric duct is heavily reliant on testosterone in order to stick around. And since we talked about the fact that our testosterone cannot act on our mesonephric ducts, we are not going to be able to support these structures. So these patients will not have any of the structures we classically associate with the mnemonic seed. Let's move on to talking about our breast development. So these patients are usually going to have some degree of breast development, and this is because there is no effect from testosterone. So we have an imbalance of the effects of estrogen and testosterone. So the estrogen that is present in these patients is going to go ahead and encourage breast development here, and we're not going to have any testosterone to counteract it. Lastly, let's talk about our absence of pubic hairs. So we know that it's going to be androgens that lead to the development of pubic hair, and this condition is all about androgen insensitivity. That receptor cannot respond to those androgens. Therefore, these patients will have very sparse or very little pubic hair, if any at all. In terms of diagnostic workup here, we're going to have a karyotype that is XY, and then we're going to have increased levels of luteinizing hormone as well as increased levels of testosterone and increased levels of estrogen. Now, why is it that we have increased luteinizing hormone? And it's because the receptors responsible for sensing testosterone in the pituitary are not working. So the pituitary cannot sense that there are extreme levels of testosterone around, and so it keeps pushing more and more luteinizing hormone in order to try and get up from the absence of testosterone that it thinks it's experiencing. And why increase estrogen? Well, the change of testosterone into estrogen that occurs throughout the body has not stopped happening. So we have an excess of testosterone, and this is going to switch over to be converted into estrogen. And this is what allows us to develop things like breasts in these patients. Let's wrap up by mentioning that this is X-linked recessive in case we get some genetics questions on our exam. And then let's do a quick high yield review. Remember, in terms of 5-alpha reductase deficiency, it is all about DHT, and DHT is going to be responsible for development of the external male genitalia. And so we're going to have errors in that development. It's important to know these important embryological structures and also know that upon puberty, things will start to change when we get that big boost in testosterone. Moving on to androgen insensitivity syndrome, remember, we are also going to have an XY karyotype here. It's all about a defective receptor and its inability to sense testosterone. This is going to lead to failure of maintenance of the mesonephric ducts. However, we still have all the normal structures of the testes, so our production of AMH is going to lead to regression of the paramesonephric ducts and all the associated structures. This is going to lead to things like primary amenorrhea as well as a rudimentary vagina. However, the excess of testosterone in these patients is going to be present. However, it will not have any effect, so we do not expect these people to have pubic hair, and additionally, that testosterone is going to spill over into estrogen, which is going to lead to things like female breast development. It's important to know that this is going to be X-linked recessive. And then, as always, always refer to our diagram because it's going to guide all of our thought processes and allow you to get third order questions right when it comes to embryology on the exam.